So today I'm really excited to talk to everybody about translational digital biomarkers in drug discovery and development. This is the first part of actually a four-part series on these technologies presented by the North American 3Rs Collaborative. And today we'll be just giving an introduction, talking about opportunities, challenges, and practical implementation. As a note, so we are using MS Teams webinars for this platform, so please submit questions via the chat. If you particularly like a question and want that question in particular to be answered, please just thumbs up it in the chat, give it a reaction, and we will be answering those at the end of the webinar. We'll have about 30 minutes. And just a reminder, we'll have all the attendees be muted and have their cameras off for best flow. So first, really brief introduction to the North American 3Rs Collaborative. We are a nonprofit whose mission is to advance science, innovation, and research animal welfare. And we do this through facilitating collaborative 3Rs initiatives. This is a picture of our leadership team and our board of directors, and I hope that you can appreciate the diversity of different institutions that they hail from, not just academics, end users, CROs, government, and other nonprofits. And our strategy is to identify initiatives that we believe have strong scientific evidence to support their use, will ultimately make a big impact in the field, and importantly, have real-world practicality. We currently have 6 key 3Rs initiatives focused on rodent health monitoring, microphysiological systems, translational digital biomarkers, refinement, creation of a 3R certification course, and compassion fatigue, resiliency. And with that, I'm actually going to turn it over to our presenters. We have 5 wonderful speakers today. They're going to walk you through kind of an introduction to these technologies, challenges, opportunities, going through introduction, evolution, technical requirements, practical use, and then finally, I will be moderating the Q&A. And with that, I'm going to turn over the screen sharing to Shapan, who's the lead of our group.
Thank you so much, Megan, and welcome everybody. Thank you so much for joining us here today. So I'm just going to give a brief introduction to the topic, and then other colleagues are going to jump in and talk about various aspects of these technologies in more in depth. My name is Stepan Baran. I'm currently a Chief Scientific Officer at VeriSim Life. It's an AI machine learning platform for decision making in drug discovery and development. But before I was with this company, I was also at Novartis, leading emerging technologies and having an opportunity to run decentralized trials on a clinical side, as well as working with these specific technologies preclinically. So I also just wanted to remind you that this is a 4-webinar series. This is our first webinar, so it is an introductory webinar here. And then we're going to have a second webinar, talk about more about the technology and how to engage with them and challenges in the processes. And then also third and fourth webinar, where it's going to be even more of a discussion and engagement so folks can ask various questions regarding these technologies. So before I jump into talking about the digital biomarkers, why are we looking at these different solutions? And I think many of you guys know that the drug discovery and development journey is treacherous. We have really challenging path to get medicines to patients. We start off with quite a few compounds, and it takes, about 15 years and $2 billion to get something to our patients. So there's definitely opportunities to refine that process. And then so the other aspects here is, the translation and the clinical relevance, but also the novel candidates or novel technologies that are available to us and to our patients, and they require novel models. Then we also have patients. We have smaller populations of patients depending on rare diseases, for example. We're also looking at pediatric patients and sex, ethnicities. And before, when we're looking at medicines, we're looking at one medicine for the patients, but we know patients have multiple diseases. So how is that one medicine going to interact with other medicines, right? We need models to predict that. And some of the in vivo models are not necessarily relevant or not even possible for these novel medicines. And then also we step into the personalized or individualized medicine. So there we need to be able to predict what happens with patients as well. And we need novel models for appropriate models for that to predict that. So there's various tools that we have. We utilize AI and machine learning, for example, microphysiological systems. But today we're going to concentrate on the digital biomarkers. So the North American 3Rs has various initiatives, and one of them is the Translational Digital Biomarker, which this presentation and the manuscript that we're going to be talking about came from that initiative. And the idea here is to, in pre-competitive space, in a collaborative aspect, increase the adaptation and regulatory acceptance of the translational digital biomarkers that can refine and reduce animal use in research. So we have currently 13 members, tech providers and end users. There are some examples here, and we hope that if you're not a member currently, that you would come and join us. And you don't have to have a lot of experience with those technologies or even at all. But you can learn from other folks that are part of it. And if you have experience, again, it would be great if you could join us. So we collaborate to really establish an improved understanding of the value of these technologies and also facilitate optimal implementation. As with any technology, there's a lot of aspects that come along with it. Some of those we think about upfront, some of them we don't. So here's an opportunity to kind of learn from other folks that already have engaged with those technologies and are engaging. And then also, how does technology can help us promote the 3Rs? And there's definitely a significant impact, and I think you'll see through this webinar and following webinars how we do that. So this initiative, that Translational Digital Biomarkers, has 4 areas. We have educational, technology showcase, connections, and regulations. And so here, from one of the aspects is the education. So we have published a manuscript that's actually one of the deliverables that we had done within the last 12 months with how to engage with these technologies, and we're going to continue to evolve that. We also have an educational resource page and also regular posts about different evolutions of these technologies and publications. And again, this is in development and in progress. We also have presented at 5 different conferences and webinars, and we're going to continue to do that more and more, as you can see, even with this webinar. We're also going to have a tech hub, which is being evolved currently, where you're going to be able to come in and see various technologies in this area that are available and information regarding them. So the things we're going to be talking about today are also in the recent manuscripts that we're able to publish, and you can see that the authors there. It's an enormous collaborative effort. As you can see, we have technology providers, end users, regulators, and others. So, while we're looking again at this technology, I kind of touch on those topics a little bit, but over the last 60 years, the way we assess animals has now really evolved. And I'm not going to jump into too much details because Lucas in the next section is going to provide you with historical perspective of these assessment tools. But one of the ways that we do that for assessment, but also even for studies, we remove the animals from their home environment. And there's enormous amount of studies demonstrating that has an impact on the animals. Also, when you have 2 different or 3 different people assessing the animal, they have different interpretations of what they can see. So even just think about assessing how dirty the cage is, and have 3 people go in and see if they come out with same results. So this technology, digital biomarkers, allow us to remove that the individual is biased from assessing our animals. So the other big part is looking at, right, in clinics in patients, what are we doing with patients, and through reverse translation, bringing that back to us. So obviously, traditional studies in clinics, as where patients come into the clinic, they're assessed, they can even be instrumented with various technologies there. But that's challenging because they have to go into the clinic. So for example, a couple summers ago with my kids, we were traveling through United States and traveling through Wyoming, coming to one of the hospitals, it can take 5, 6, 7, 8 hours for the patients to do that. So very often they might not be able to participate in the trials. And the two biggest challenges with clinical trials is patient recruitment and then retaining those patients on those trials. So over the last few years, we have invested enormously in remote ability to monitor patients and address patients at home. So we bring a trial from the clinic to the patient's home. And it allows higher recruitment of patients, retention, but also more objective assessments of those patients. So it's the same thing with the animals. Instead of taking the animals outside of their home environment and assessing them and running studies, now we're able to bring that study to the animals, to their home, and assess them there with these technologies. So these digital biomarkers allow us to assess or provide a holistic view of the animal. So on the bottom here, you see a conventional assessments, right? We look at the animals once, maybe twice a day. I know there are exceptions, but really, 95% of the time, when the animals are housed, we don't really assess them, we don't look at them. So we miss potentially information that we could extract from observations of those animals. And digital biomarkers provide us with this continuous monitoring. So on the top there, you can see kind of the white and the gray. And the white is monitoring the animal activity during the day, and the gray is during the night. And as we know, rodents, especially mice and rats, are more active during the night. And you can see that here. And typically, we don't monitor them during the night. So again, these technologies allow us to monitor them 24/7 continuously and have this holistic view of the animal. So as with any technology, you really need to start having the conversations, especially with different stakeholders, and a meaningful conversation. You have to be aligned on the terminology. And so we have spent a lot of time doing that. And again, I'm going to go through some of the definitions, but we have to remember that this technology evolves, and its definitions will also evolve with the technologies. So what's the digital biomarker? So a digital biomarker is the data collected continuously from unrestrained and uninstrumented animals in their home cage environment. So these animals should not have undergone minor or major surgery, with the exception of RFID chips injected subcutaneously. And the reason why we have that exception there, because we know that regulations and definitions for minor and major surgery differ in, for example, in North America versus in Europe. So then what is a translational digital biomarker? So that's an objective, quantifiable measure of physiological and or behavioral response to a disease progression or a therapeutic intervention that is collected by means of digital monitoring technologies, including both internal and external sensors, which is clinically relevant and translate between preclinical studies and the clinic. So then, how do we define home cage or home environment? So that's cages and environment where animals are housed for the majority of their lifetime in a vivarium. And benchtop cage technologies are cages and technology typically used for experimental test environments. They're not designed for permanent housing, but where animals are housed for a short period of time to run these studies. And again, those type of technologies exist. They have an enormous impact on drug discovery and development. But here we're talking about home environments. They're also scalable. Now, what do we mean by scalable? So scalable, meaning that we have the ability to monitor hundreds to thousands of animals within their home environment. So also validation and verification of these technologies, right? Because you have engagement with the hardware, the software or the platforms, machine learning and AI that comes along with it. So these terminology also align from our clinical colleagues, where these similar technologies are being utilized to monitor patients. So analytical validation entails evaluation of data processing algorithms that convert technology collected measurements into outputted metrics. What does that really mean? So really is that the algorithms that are being utilized to extract information are really working appropriately? Are they showing what we want them to show, what our expectations are? And then technology verification. So here ensuring through demonstration of precision, reliability, and reproducibility, the device is measuring and storing data accurately. So again, if you're expecting something to be measured by the device, making sure that the device can do that over and over and over again with a similar output. So again, assessing the precision, reliability, and reproducibility. And then we have the clinical validation, which is accomplished by demonstrating that the technology adequately identifies, measures, or predicts a meaningful clinical, biological, physical, functional state or experience in a specified animal cohort and context of use. So here we're demonstrating that this is appropriate for us to utilize for our studies, and the information it's providing later on being translated into clinics. So now I'm going to pass it on to Lucas.
We still need you to be unmuted, Lucas. Yeah, there you go.
Thank you very much for the introduction, Shapan and Megan. I see my title slide. I don't see a video inset, but I hope you're seeing the video.
So we don't see your slides yet. You may need to stop presenting, share the slideshow, and then restart presenting.
How about this?
Yeah, looks great. Thank you. You're good to go.
Okay. I don't see video, but I do see my title slide. Okay. So Siobhan gave an excellent introduction into our ambitions, what we want to achieve as a group and as a field, where we're moving towards. And I'm going to jump back in time to give an overview of a very brief evolutionary overview of what is becoming digital biomarkers. What are becoming digital biomarkers according to the definition that Siobhan just gave? My name is Lucas Nolders. I'm the founder and CEO of Nolders Information Technology, a company headquartered in the Netherlands that develops technologies for the measurement of animal behavior and physiology, focusing on animal behavior and in particular rodent behavior. And this is a summary of my presentation. This is a very dense graph that shows the evolution of the measurement technologies for animal behavior in this central section here, this part, and animal physiology, and the integration of these technologies eventually in housing systems, the rack-mounted cages that we find in the vivarium. Along the x-axis, you see timeline from 1980 to 2020. That's an arbitrary period that I can oversee relatively well. My personal involvement in this field started in, say, end 80s, early 90s. So this is the period that we'll be covering in this 15 minutes. There's no time to discuss everything in detail, so I will be a bit selective. I'm starting with this section here, the animal behavior, starting in the 1980s, and I'll illustrate these various elements in the slides that come. So in the 1980s, the rodent behavior was roughly measured according to, say, two ways of working. One was the operant paradigms, operant chambers, also Skinner boxes, which measured the interaction between an animal and devices. The stimulus presentation devices presented cues to the animals, and the response of the animal to those conditioned or unconditioned stimuli was measured. You see here this cage with a variety of devices connected and the electrical wiring, the reading the signals and providing either positive or negative reinforcers, food rewards or mild electric shocks. Clearly, the animal cannot show, say, exploratory behavior in this. There is no video connection. Nothing of the behavior is recorded. Only the responses are recorded. These operant chambers existed until today. They have evolved with a continuous development of new stimulus devices and response measurement devices. Multiple companies in the world are making the extensive portfolios of operant chamber equipment. The other way of working is the study of spontaneous behavior of animals in open fields or Morris water maze, in which the animal navigates towards a hidden platform in a Morris maze, or you record whether the animal is walking along the wall or in the center of the arena. Originally, these recordings were done manually by human observers without any recording equipment. That all came later. This type of mazes and open fields have evolved with a growing, the additions and extensions of different devices to measure specific aspects of brain function, such as the elevated plus maze, the Y maze, the zero maze, the three-chamber maze, and many more. But none of these devices that you see in the picture here qualifies as a home cage. The animal is taken out of its home cage, placed in this arena, measured during a short period of time, and then taken back to the home cage. And as Shapan explained in his introduction, that has disadvantages. And around the beginning of this century, the various groups decided to develop novel concepts whereby the experiment is taken to the animal rather than the animal taken out of his home cage to the experiment. And in some 20 years ago, that led to the development of instrumented home cage-like test cages. So these are not real home cages. They are labeled as benchtop instrumented cages in Shapan's definition, or in our group's definition. And here are just two examples. To the left, you see a cage called IntelliCage, in which many mice can be monitored simultaneously, whereby the primary sensing technique is RFID, the chip that interacts with the sensors in the corners, where the presence can be recorded and the interaction with food or water supply for the animal. And for all animals in a group, this measurement can continue for hours or days on end. Another approach is taken by this one here, the phenotyper, which I'm involved with personally, in which one or two animals can be tracked with a video camera, and the spatial behavior and exploration is recorded for hours or days on end. Again, these are not genuine home cages. They're not rack-mounted home cages. They don't have the scalability of home cages, but this is where we started 20 years ago. And both technologies have evolved with the addition of new sensing and sensors and response devices, and more software for the automated behavior recording. The behavior recording in these systems is originally or traditionally and still mostly based on video. Video started with video recording in the 80s, just a camera above a cage, and in the early 90s, software-based video tracking was invented, whereby a video image is transferred from an arena to the computer, and the digital image processing software tracks the center of gravity of an animal. And later, the different body points were added, nose point and the tail base and contour. And based on that, the simple rules for automated behavior recognition, ABR, could be created. For instance, by measuring the distance between the nose point and the tail base, you can define a rule that detects the stretched, tense posture of an animal when this distance exceeds a certain threshold. That is very simple automatic behavior recognition. And later, neural networks made their entrance, and Bayesian networks and Bayesian interference allowed us to feed lots of features from the digitized object into a classification algorithm, allowing us to automatically distinguish between a range of behaviors in a rat or a mouse from an overhead video camera. And this is what is happening in the benchtop cages I just showed you. And more recently, deep learning techniques have arrived or are now widely adopted through which allow us to obtain a much more, even more accurate classification of the behaviors, or the pinpointing the detection of specific body points, such as the nose point. Here we see the nose point of a mouse in a cage. With traditional contour-based tracking, the nose point can easily be confused with the black walls of the cage, meaning that the point is not allocated on the real nose point. With the deep learning techniques have allowed us to, even in these very challenging circumstances, put the nose point exactly on the nose of the mouse, which, for instance, in object recognition task is a very important metric. Now, these so far, a quick run through behavior measurement and video. Let's move to physiology. With physiology, many techniques have been developed to record the cardiovascular measures. That's the line up here, the gas exchange, food and water consumption, and metabolic measures. No time to discuss them all, so I'll currently limit myself to the cardiovascular measures. And that started in the 80s when the tail cuffs were applied to rats placed in restrainers. Here we see a rat where you hardly see the rat because it's inside a restrainer, but the tail cuff, the cuff around the tail, allows the researcher to measure blood pressure. Well, clearly, this does not qualify as measurement of refinement in terms of allowing the animal to move freely. That became possible in 1984 when data scientists introduced the wireless telemetry with a implantable sensor and a radio transmitter, which picked up biosignals from inside the body of the animal through leads connected to arteries or organs, and transferred that signal through a receiver placed under the cage and to the computer. So that was really a breakthrough, and it has become a widely adopted method. And over the years, that technology has become more smaller, allowing mice to be implemented with the telemetry. So in the 90s, and even more recently, the radio technology has moved from analog to digital, allowing multiple animals to be tracked in a cage environment or monitored in a single cage, whereas the analog technique allows only one animal to be monitored in a cage. The digital allows group-housed animals, which also complies with animal welfare ambitions. And also these digital telemetry devices have become smaller and smaller, allowing multi-animal tracking in mice. And with switching to higher radio frequencies, it is no longer necessary to place the receiver plate under the cage, but at a distance from the cages, so that multiple cages can be monitored with a single antenna. And by doing so, this has led to solutions for a variety of biosignals such as ECG, heart rate, EMG, muscle contraction, EEG, brain signals, blood pressure, and more. So back to the index page, I've reviewed the behavior. I gave you a quick run through this line of the physiology. The time does not permit to go into details about these other, the metabolic and gas exchange measures. Let's now move to the housing, because eventually our dream is to build this technology into home cages for continuous measurement in the vivarium. Now, home cages traditionally or conventionally are cages in which animals are placed in an enriched environment, and these cages are then placed into racks. And the original home cage of the 80s did not measure anything. It was simply a housing environment in which animals are residing and spending their laboratory life. And also, it's not obvious how to do measurements in this home cage, because if I put a video camera above this cage, the way I showed it in the benchtop environments, then we'll be seeing this grid and the food, and the water bottle blocks the view of the animals. And that became even more challenging as in the 90s, the individually ventilated cages were introduced with even more objects in view blocking overhead cameras. So there was no obvious solution on how to go about the monitoring animal behavior inside the rack-mounted home cage. But as the benchtop systems evolved in the beginning of the century, that stimulated also the development of rack-mounted home cage technologies for automated behavioral and physiological monitoring. And here we see an example of that, a home cage with the capacitance base plate. It's called the DVC cage, whereby this plate contains capacitance sensors that can record, say, roughly record activity levels of animals inside the cage placed on top of this plate. And no individual recognition there, but the activity measurement and also the measurement of the humidity of the bedding. A more recent development is the addition of RFID sensing, with RFID sensing plate placed dimensions such that it fits in a home rack under the home cage, whereby even the body temperature can be retrieved from special RFID chips that have a temperature sensor added to them. And then there's developments whereby the video cameras are placed next to the cage, so sacrificing a slot for a cage in the rack, but having a system that can record the behavior of individual animals inside the cage. There's all sorts of challenges there with respect to individual recognition and accurate, say, recording of movement, but it's a really interesting step forward, which is also, say, represented in our team, in our group by the company Actual Analytics. So here we have company UID, Techniplus makes this, and then the technology made by Viam is another approach whereby video is even used to capture physiological signals from the animals in a remote fashion. So these are fascinating developments. Many of them are still in their, say, early stage developments of the past 5 years, but there's really a fascinating future ahead as these technologies become more mature, more robust, and as video imaging and AI techniques allow us to get more data out of the sensors that we can mount and add to the cages in the rack-mounted systems. So please stay tuned for these developments. Put your questions in the chat, and at the end of the webinar, we will answer them as well as we can. And I now stop the sharing, and I give the floor back to Megan.
Awesome. We'll turn it over to Stefano.
And I'm going to start my sharing. Bear with me.
Awesome. And Lucas, if you want to turn off your camera, you can.
Can you see my screen?
Yes.
Brilliant. So my name is Stefano Caburo. I'm the Global Scientific Director at Techniplus, and we develop, along with cages and other things for the vivarium, also the digital ventilated cages that were shown before. But today I've been asked actually to talk about what are the technical requirements and the challenges of study design. I will focus specifically on the study design and some stats that we have been identifying also in our paper. So it's a joint effort, as Shepan said before, because data or digital biomarker, digital translation biomarker that comes from the home cage are in 24/7 manner really require a lot of, depends on the source that you're basically working on. It requires a lot of effort. You have the IT and computer science to make sure that the data flows, if the data on the cloud or on local machine. You have the biologists basically asking the question, and sometimes they don't know because they have completely novel data. And on the other side, you have biostatisticians that basically help addressing this question. So it's really a joint effort to go through this data. And this is, coming a bit away from the traditional way of analyzing data. I'll show you later in a second. So a small agenda, especially for study design, we have to keep the numerosity of the cage in consideration if you have 1, 2, or 5 animals per cage. Light and other environmental conditions are very important. As Lucas mentioned before, you might have, material bedding, you might have tubes and other stuff that might, for instance, interfere with the vision of the camera. The sleep status of the animals is a real predictor of behavioral outcome. And mixing group, transgenic, wild type, or drug and not treated, and emotional contagion is a really brand new thing that came out very recently regarding study design and data analysis, parametric versus non-parametric data. So numerosity. If you have 1 animal per cage, you can actually really look at several behavioral expression along with distance travel, grooming, very complex behavior. You can easily detect it with several technology and apply what Luca described before. It's also easy to compare with historical data because typically animals are analyzed one after another in group, but in an open field, elevated plus maze, you name it, you have one animal per elevated plus maze or per open field. Unfortunately, there is no group interaction. So for instance, if you have a model for autism, where you will evaluate what is the contacts of the two animals, you will miss it. And also importantly, mice, but typically rodents, for long study design, like in a home cage, they will suffer for cold stress. So mice like to hug, and also other rodents. And where we keep them not at thermal neutrality, like 27, 28 degrees, but at 20 degrees, they will have cold stress and therefore move more and have higher metabolic rate. When you have animals or groups that are higher than 1 per cage, the group activity is much more representative for such studies. Depending on the numerosity, 2 or 5 animals can actually hinder some behavioral expression because animals might overlay, you might not see it completely. And also you might take into account the hierarchy that might be established before, especially if you use animals that don't know each other. It might skew the data. For instance, if you have a running wheel, it happened in the past that the dominant will be using the running wheel and the other 4 will not be using it. Environmental condition can impact very heavily the recordings. And that condition might be hard to be constant across several days. And the video alternative, of course, can be used infrared cameras, which is irrespective of light. But the room light can actually affect the behavior. And this is an example here with a rack, and there were light just next to it. And depending where the cages were actually taken, if you're on the top or the bottom, the activity actually can decrease or increase by 15-fold in this direction, 3-fold in that direction. And it was shown actually in this example here. In this graph, you have here the Zeitgeber and then the activity, whereby the bottom cages were having much more activity as compared to the middle, and the upper cages so close to the light. So environmental light might really affect the behavior. And if you do a study design that respect, you might want to have all the same level or too closer to each other. The environmental enrichment can also impact the recordings. Environmental enrichment, or EE, is mostly mandatory, especially in Europe. That's where we are. And we have home cage conditions are actually needed to induce welfare. However, tubes, paper, and as material, mouse might, again, as we said before, hinder the vision of specific behaviors and behavioral interaction. The voluntary also running wheel that can be used as a toy might actually modulate spontaneous behavior and eventually neurochemistry. Is actually addictive. And the suggestion is actually to keep the environmental condition constant across the experimental cages. So if you have, let's say, 16, try to keep the environmental condition constant, or environmental enrichment. There was a recent paper by Tam and PNS 2021 that basically was saying that the sleep performances of the mice can be a predictor for the behavioral performance. And changing in the circadian rhythms distribution might indicate changing the welfare status of the animals. And like in this case, if you have true home cage monitoring and remote monitoring, identify an hypo activity of the animals, you might not want with that cage be running anything which is based on locomotion, for instance, because it might actually affect your output, or you can take that into consideration and actually correct your data. Mixing groups was a defect of the emotional contagion, and which is nothing else than empathy. So let's say that you have a study case in which you have subject which have an unknown mutation, or you have a drug X, and you want to study behavioral expression. And the recent studies are called emotional contagion, which is basically the transfer of a specific emotion to a conspecific, has basically shown the following: that both mice and rats alone show comparable reduced contagion as compared to animal tests in the group. In essence, if you have something that would induce an hypoactivity or change in the behavior, it might actually affect, and it's in the the transgenic, it might actually have an effect also on the wild type if they're kept in the same cage. This is the review, and it might have actually potential effects again on your study design. And lastly, with regards with data analysis, the on-cage behavior does not typically follow parametric assumption. And t-test, ANOVA, which I think 80% of non-biostatisticians are used to use, are actually parametric tests. And the data coming out of the home cage behavior are typically skewed, do not follow normal distribution, and they are intercorrelated, which are the three assumptions which are used for t-test and ANOVA. And therefore, you will actually violate the assumption if you were to use such tests. Instead, the use of non-parametric analysis actually is being suggested. And that's why, coming to the first slide, collaborating with biostatisticians will help to analyze the data. One example is actually to use 1 or 2 weeks baseline, as I will show in the next slide, would actually mitigate these effects. And because the typical home cage data is very variable. And in this case, for instance, in which it was an example of acute colitis induced with DSS, in which the clinical score was taken, and with 2.5 DSS, you have the highest value in clinical score. And percentage of activity, of the baseline activity of 2 weeks was taken, you can see that the reduction in the activity very nicely corresponds to the increase in the DSS. So this might be a way to analyze statistically and parametrically the data. And with that, I would like to conclude and leave the stage to Sean and Laura. Thank you.
Awesome. Thank you, Stefano.
Let me share the slides. Let's see. Give me a second. All right.
Awesome.
Yep. Thank you, Megan. How about it, Laura?
Thank you. So I'm Laura Shavitz. I am, just a little bit of background on me. So I was part of, or I spent quite a bit of time working with these digital technologies as part of Vium, which, as you'll note, I put in the chat. Vium was actually acquired by Recursion, which is where I am now, where we're using these types of technologies on a regular basis in our own drug discovery pipelines. So what I'll share with you today, along with Sean, is some of our thoughts around practical use of these types of technologies, and asking some of the, or figuring out some of the questions that we can answer with these technologies. Sean, do you want to quickly introduce yourself before we get into it, or?
Sure. And then I'll skip over my slide later. Hey, folks. So I am a comparative and translational sciences veterinarian at GlaxoSmithKline, and I've been working in this digital biomarker space with many of the folks you've already heard from for the last couple of years.
Perfect. So you've heard quite a bit now from Stefan, kind of setting up why digital biomarkers, how we think about it in terms of definitions. You heard from Lucas about kind of the evolution to where we are today, and with Stefano about some of the some of the considerations around how to think about how to apply these digital biomarkers. What Sean and I want to talk about are really the practical implications of them, and what types of questions you should be thinking about answering with this type of technology. So what I'm showing up here is a very busy slide with all of the potential reasons why you might want to use translational digital biomarkers, from improving animal welfare. Can you reduce morbidity, mortality, operational, the ability to do remote animal welfare checks, for example, and reduce the number of animals that are potentially found dead in cages, improving your scientific and outcomes for studies. Can you identify better, objective, quantifiable ways of measuring animals on studies? And even strategic. So in drug discovery, we often think about how do we get drugs to patients faster? Well, if we can move through our animal studies more quickly, get earlier decisions that we think are more clinically relevant, that's a big win for both the companies that are developing the drugs as well as for the patients who are ultimately going to hopefully benefit from those drugs. And so, there are lots of reasons why you should be thinking about the use of translational digital biomarkers. Sean, can you go to the next slide? But the big question always becomes, well, practically, how do I translate that into thinking about what are the right studies that I should be using these digital biomarkers for? And this is often where both internally within our own companies, as well as talking with others across companies, we fall down, which is to say, well, how do I know whether or not a digital biomarker is going to be useful on a particular study? And so what I've highlighted in this slide as a kind of a beginning brain teaser is a way to think about whether or not studies you're running could potentially benefit from the use of a digital biomarker. And so what I'm showing you here are 1, 2, 3, 4, 5, 6, 7 or 8 different bullet points where we've already found that digital biomarkers can help us improve the quality and the outcome of studies. And so what I'll do is just quickly run through each of them and give you a small taste or example for how we've thought about the use of digital biomarkers in these contexts and where we found them relevant. And then at the very end, I'll just give you a few recommendations around how to think about it in the future. So the first is around measures or traditional measures that you might find collect on a study in which they're not necessarily predictive, perhaps they're not reproducible between studies or labs, or more importantly, may not translate to clinical outcomes. And so one great example here is one that we actually use quite often at Recursion, which is idiopathic pulmonary fibrosis. For any of you who have worked with a model like this, you'll know that it is very challenging to reproducibly introduce the lung inflammation and the lung fibrosis in these animals. And so using digital biomarkers helps us to assess how well we've induced the disease in the animals before we even get to the therapeutic outcomes. And so it helps us actually make sure that we are, even if we can't fully reproducibly introduce it, it gives us a baseline for understanding the response to therapies based off of what we can actually track longitudinally across the study. In the second bullet point, measures that require extensive training or a large number of animals per group for statistical significance or even pain and distress. We've published an article, or there was an article published quite a while ago, back in 2017 now, on using digital biomarkers for arthritis. Arthritis, as many of you know, caliper measurements is a traditional way of knowing whether or not your animals have been induced with arthritis. that requires extensive training and actually there's a lot of variability between measurements for different animals and for different experimenters. And the ability to use digital biomarkers can actually reduce that the challenges that you will get between measuring that paw thickness between experimenters. And so, as well as we can actually potentially stop studies earlier before animals get to significant pain and distress, because we would potentially get more sensitive measures. Longitudinal disease tracking. This is another great example where there are some diseases where there's no outward sign. So we've used some of these digital biomarkers to look at like lung metastases, for example, and the ability. What we'll see is increased breathing rates as animals' lungs fill up with lung mets or tumors. And that's a great example where it's very hard to actually measure that over time unless you have some sort of kind of physiological output. Models with unexpected mortality. We often find, or we've found in several instances, that there are models in which you just cannot predict when animals are going to die. They appear normally to spontaneously die. And the ability to track a lot of these digital biomarkers, you can actually find subtle signatures in those digital biomarkers, which would allow you to identify and then prevent the death of those animals. Also, measures that are sensitive to the emotional state of animals. Stefano talked about a few of those, but also knowing that procedures, even just handling animals' blood draws, etc., can have an effect on animals. And so if you need to remove those confounding factors, digital biomarkers can potentially help you do that. Situations where there are modest therapeutic improvement, or where you have to really hammer a model in order to be able to distinguish disease from healthy controls. Digital biomarkers are often more sensitive and can allow you to subtly identify therapeutic effects without having to hammer those animals. And I think a perfect example there is some of the work that's been published around the paraquat fibrosis models as well. And then the last one before I turn it over to Sean are models where baseline behavior or physiology can impact disease induction or variability. And here, potential examples would be around circadian rhythms in the immune system. We know that those are tightly regulated. And so if you've got animals that are not on kind of a normal circadian rhythm, you may actually get quite different induction if you're looking at immune models. And so these are things to kind of think about, potential questions or challenges that you might have, and how digital biomarkers could potentially help you solve those. What I'd recommend for everyone, if you don't have systems in-house, we have on our website, as Stepan highlighted at the very beginning, a lot of resources and studies that have been published. Take a look at what's on the website at this point. I would say that many different models and the potential use of digital biomarkers in those models have been published. And it's quite likely that a model that you're interested in or something that is very similar, you can already find information on whether or not digital biomarkers would be relevant or useful in that animal model. And then as you think about bringing in technologies to be able to monitor those, we would highly recommend that you do an initial pilot study just to see if you can distinguish controls from your diseased animals, just using very standard or using these digital biomarkers. And then after that, follow up with your standard of care compounds, as well as any compounds that you'd be potentially interested in once you know that you see that signal. So those are just a few kind of practical as you think about designing experiments. Sean, over to you.
Excellent. Thanks so much, Laura, and a warm welcome to everybody who's joining us. Right. So as the other talks have hopefully set you up for being, super excited about, I can't wait to bring these technologies in and start using them, and being able to generate some incredibly valuable, monumental context of use studies. How soon can we do this? And so just to give everybody, a chance to take a breath and realize that, from that moment where your institution has made the decision to bring this technology in, that you're looking at, probably at least 5 months, potentially longer. Some of that, right, depends on the complexity of the technology, right? Where is it on the kind of plug and play spectrum versus, requiring a whole lot more in terms of both setup as well as training. And so, as my colleagues have highlighted, right, there are lots of things to consider. In the remaining 4,000 minutes, I'm going to cover everything, right? And so I say it that way to point out that there's lots and lots to think about. I'm not going to cover everything, and I was just told that I only have about 5 or 10 minutes and not 4,000. So with that in mind, I have 3 slides that I'm going to use to highlight some things that we've seen as challenges, and some things to consider as your institution thinks about, how you're going to solve those challenges, and get to a point where you are able to use these technologies to collect novel and valuable data. So depending on the specific technology, right, there is that will inform the degree of information technology support that you require, and certainly anything that is dealing with a huge amount of data, right? And so certainly when you compare these technologies to most things that are already happening in the vivarium, we're talking orders of magnitude more in terms of data. And especially in cases where that data needs to go out from your institution to a cloud somewhere and then potentially back in, you're going to need help unless you have all of the IT related infrastructure knowledge and resources. There is a lot to navigate. And so lining up the right IT people wherever you are early on, getting them involved early on is absolutely critical. And you should plan on being in fairly frequent contact with them as these things get up and running. Some of that is related to cybersecurity, right? And so we have the there are 2 different pulls going on here, right? All of our institutions need to protect our internal IT platforms, and at the same time, right, we want to be able to quickly and easily access any of the data that we generate. And so if your institution is like mine, right, there are lots of processes to work through, and just to recognize that early on, and that it's likely going to take a bit of trial and error. As you look to bring new technology into the vivarium, you got to think about where you're going to put it, right? And not just the caging itself, but any of the related hardware. How is this going to be cleaned? How are the rooms going to be cleaned? Do you need to do anything different than what you're currently doing? Do you have the opportunity to do something different based on how you're using the technology? You will also be talking quite a lot with whatever vendor that you're working with. And again, this is all part of the process of, learning in your own hands how this works, and figuring out all of these small but critically important steps to get the technology up and running. One of the things, another thing to consider is how using the technology in a model that you're already very familiar with may actually impact the model itself. And so, right, one specific example of that would be, right, if you're adding a wheel into the home cage in a model where you previously haven't, right, maybe you want to get a sense of how that wheel running changes over time. There's likely to be a metabolic impact to the animal, right, compared to an animal that's that has not had the opportunity to do that wheel running behavior. Similarly, something else to think about is that because most of these technologies offer real-time or near real-time monitoring, right, you have the ability to detect a change where you couldn't previously. And so, to think through how that's going to impact how you run your model, right? And if you get this health alert at noon on a Monday, do you respond to it the same way that you would at 11 p.m. on a Saturday? Depending on the technology and what you want out of the data, right, there may be some decisions to be made about your housing density. And if the technology is such that it gives you a readout on the cage level, right, which scenario, right, single housing versus social housing is the most valuable for your model, and how do you appropriately weigh that with the ethical considerations, where we know that in general, right, social housing is probably one of the most important things that we do for our animals. You're going to generate a huge amount of data, right? Think carefully about how that's going to be handled, how it's going to be retained. Do you need to retain the raw data for the same period of time as you might any of the algorithmically generated endpoints? I shared with you a theoretical timeline at the beginning of this, right? And it's absolutely appropriate and smart to share that timeline out with the relevant stakeholders. Recognize that it is very likely to change, and that's okay, right? That's all part of the process of bringing something novel on. But then to also make sure that you are communicating that out so that folks don't ask, what were the results of the study that actually hasn't started yet. Verification validation was touched on a bit, right? And conceptually, as a pilot study, great place to start is to think about how you can do your traditional model with traditional measures with adding these digital biomarkers. And that from that pilot study, you'll be in a much better and more experienced and knowledgeable place to plan your next studies. Some of these technologies allow for a very different thought process in terms of study design. Laura talked about that a bit, right, with the idea that you could have specific digital biomarker-based enrollment criteria, right? Again, similar to what's done in the clinic. I'm not going to touch on the regulatory bit for now, right? And so all this stuff is new, and it's fascinating, and there are so many shiny things to look at, and we are also talking about changing the way that things are done, and it's just important to recognize where people might be in terms of their appetite for change, and to think about, how you might move your colleagues to a place where they are more eager for it. The use of these technologies, I think, provides a tremendous opportunity for collaboration, and both from the perspective of within a particular institution, right? Often these technologies are brought in for a specific reason, and then other folks hear about it and can quickly see how other groups or other concerns might be able to be answered by using the same technology, maybe even within the same study. And then what you've just listened to for the last hour or so is also another fantastic example of the collaborative opportunities that the use of these digital biomarkers presents. So I will catch my breath and stop there. And I will try to unshare my screen.
Awesome. Thank you, Sean. And thank you to all of our presenters. Can I go ahead and invite all presenters to go ahead and turn their videos back on? You can wait to unmute until questions come in. Yeah, thank you, everyone. I do want to start off just to kind of break the ice with a little poll. I'm going to launch this now. The question is, do you or others at your institutions currently use a home cage monitoring system? And while we're getting the results of that poll, I do encourage you to ask any questions in the chat. There is a question about recording. This presentation will be recorded. It is being recorded. You should have all had the little bar on the top of your screens when you joined. I am going to look at the recording to make sure that participant names are not shown, and so I may need to crop it slightly, but it will be likely hosted on MS Teams. We could also discuss hosting it on YouTube unlisted if people prefer that. Either of these things is very possible. And yeah, we would love to for you to share it with others at your company that may not have been able to do it live. So, okay, so via our poll, we're a bit 50-50. It looks like, about 6 people are using it, 6 people are not. Great, so I will go ahead and start with the questions. So first question is, great advance in the technology in this space. When do you think this will become a common practice in the field? For example, 3 years, 8 years? We know it's a slow field at times. So yeah, do any of the presenters want to field that question?
I mean, I can jump in. I think many of you, even the co-presenters here, I was pretty much with the idea that by now this will be a common practice, and obviously it's not, right? And I think one of the things that we're finding out that in a pharmaceutical and drug discovery and development industry, we're very innovative with the medicines, but when it comes to technology and especially preclinical and especially in vivo arena, we're very slow to adapt to change for a lot of different reasons. And then so I think it's going to take us a little bit longer for this to become a common practice. I think one of the questions also comes up, even like the pricing of the technologies. And what I compared is to IVCs, right? Everybody, when they came out, well, only, a few institutions are going to be able to afford those, right? And I think now you see it everywhere. So I think that's one aspect. And I think some of the aspects before that I brought up, or that we talked about, is, the translational gap, obviously, and the cost of medicines is just keeps rising. There was a couple slides that I wasn't able to show today, but there was a projection that by 2026, we're going to be almost doubling what we're going to be investing in R&D for pharma, right? So going from about 28 billion dollars today in 2026, right? That's 2 years to 256 billion dollars, right? I mean, if this doesn't engage, our interest here in the preclinical side, so I think it's going to take a little bit longer, maybe 10, 15 years or so. I hope it's a lot faster, but I think by then we're going to have to use some kind of different technologies to work with animals. So I'll just step off my soapbox and let others jump in.
Yeah, it's a crowded soapbox. I'm certain it will not happen as quickly as I would like it to. I think one of the challenges that I've seen is that right, in my opinion, the big wins for these technologies, it's very hard to tie it to a specific project, right? Because if you are looking at a capital budget and say, I can guarantee you ahead of time that if I spend X amount of money, this will move this project forward, that much faster. The problem is that where the technology is for me right now, I can't guarantee that, but I would feel more comfortable if I was able to say across 10 projects, I'm sure I can make a significant difference. And so in my mind, ideally, I think some of the wide scale adoption has to come, will hopefully come after there are some notable successes to point to, right? That both in cases where these technologies move things to the clinic significantly faster, or the other side, right, where it was very clear that the use of these technologies prevented something from going into the clinic, because a liability either from a efficacy or safety perspective was flagged that would have been missed otherwise.
I think if I can add a European perspective where we have a much stricter welfare, the regulatory bodies are really mandating us to do more and more welfare. I mean, Lucas can actually say from Netherlands, but in Germany, in every country, there was just in Switzerland weeks ago, something to ban completely animal research and the product derived from it. So we kind of been pushed more in Europe to actually use this type of technology if alternatives are not there. Of course, alternatives is the priority, and then animal is there. And we are going that way no matter what. and along with welfare, there'd be also an improvement on the model, which actually tied to the second part, like how can the bees impact the preclinical research. I can just pick an example. There was one study in which one customer was just doing metabolic study at the end, and then by having the home cage, they saw actually that the animals were from beginning different, and now you have to do 3 metabolic study, which is not a reduction in animals, but it's actually an improvement in the model, really. So.
Great. Yeah, thank you all for answering that question. Are there any other comments from speakers for the question of how digital biomarkers can impact preclinical research in metabolic diseases such as diabetes or obesity? Or did Stefano cover it?
Based on some recent things here, right, certainly there's a lot of interest in being able to tie some of the indirect calorimetry measures with things like home cage activity, and right, one can certainly see that being able to tie those two things together in that home cage setting in a way that hasn't previously been done gives you a very different insight in terms of the metabolic processes and things that we are hoping to tweak. I think the other areas or the other ways that some of this technology really matters in that space is that the ability to automate some of those measures in a way that requires much less in terms of either people or time makes a huge difference, say, just in throughput for those studies.
Yeah, sort of, just add to that. I think the other part also is kind of the whole aspect of reverse translation, right? So when you talk about to diabetic patients, right, what do you care about? So yes, they care about the glucose levels, right? But in reality, they care about their functional assessments, right? Are they able to walk around? Are they able to go running? Are they able to do, various things? So being able to assess those functional parameters in patients, and that's what we're starting to do, which even when you do that, you can extend your label for the medicines that you're working with. So looking back, at preclinical models, are we able to also provide those functional assessments? So here talking about, diabetes or obesity, but also, across other diseases, kind of, being a more broad application. So I think for us, because whatever we do on a preclinical side has to matter to folks in the clinical side. If it doesn't, the technology is not going to move forward. So for us, looking at what's important to the patients, what's important to the drug development side, and then, how can these technologies help us expedite the decision-making process and providing actionable insights as we move into the clinic.
Thank you, Stefan. Lucas, do you want to go ahead?
Say, many of these techniques that we say that are needed for metabolic measurements, such as food consumption, water consumption, body weight changes, gas exchange, have been around for quite some time in the benchtop type of say solutions, but haven't moved to the dense rack-mounted home cage in vivarium yet. For technical reasons, for space constraint reasons, for pricing reasons. So that the added value, the validity of these measurements, I think, has been proven in many publications using small-scale benchtop arrangements, but haven't scaled up to the level of the vivarium yet. And there, yeah, I think it's also up to the pharmaceutical companies to decide that they find the results obtained in benchtop systems or small numbers of say home cage-like test environments in racks convincing enough to invest loads of money, tons or millions, into creating such an instrumented infrastructure in the vivarium. And I think that the technology companies will be happy to work with pharma on that, but also don't forget that these technology companies are mostly small enterprises, so they're not Tesla-like companies that can move the field of electric driving forward with billions and billions of dollars behind them. No, there are small private enterprises who can make a small step at a time. So that's one of the reasons why these developments take so long in our field, because there are no billions to be spent on R&D on improving home cage technology.
Thank you, Lucas. And I do want to draw attention, we had a comment. So thanks to all the speakers. Very nice to see academic and industry coming together on the topic. And their comment was, while digitation affords standardization with labs and facilities, there's large variation between groups for the same biomarker. What Lab A calls behavioral digital biomarker will vary for Lab B, particularly for behavior. We need some level of standardization for the field for individual biomarkers. For instance, we have used ontological terms for phenotypes. MGI at JAX links these to genetic models and hosts this as a database. Let's start as a group to create a new generation of these ontological terms. This will be particularly important as we apply biomarkers to preclinical research and ultimately FDA applications. Do any of the speakers want to comment? I see some positive comments. Great opportunity. Stefano pointed out that there's also a research topics in Frontiers as well.
Can I just stress, I think before then we go into standardization and definitions, we really have to talk about metadata. What are the data? So the definition of that has to go on a searchable database that you will use basically then to look for your animals, which is controlled. Safety did it already in the past. They found control animals, they define some specifications that will be used and across actually different companies for, you know, reducing the amount of number of animals eventually, and start really to digitalize eye, mouse, whatever from beginning, really from the winning until the end, to have all the data into. But this shouldn't come from groups, should come actually from the authorities, FDA, NIH, saying these are the requirements that should be, you know, a panel of experts, task force, whatever, that say these are what we need, because then it will have to go into publication and into all of the stuff. So it's a huge project, but I think it wouldn't start from private, but rather from, I don't know, granting bodies or regulatory bodies.
Yeah, and Stefano, just to add, so thanks Vivek for raising that, right? And I think all of us here agree, right, that ontology needs to be worked out and addressed. I think one of the aspects that comes with any technologies, preclinical or even in clinical, it's always kind of, you know, like, you know, the regulators should have us, you know, move this forward. And the regulators are saying, no, that pharmaceutical companies should move it forward. It just doesn't work that way, right? I think now from both sides, right, people are more collaborative. But I think, again, if we just wait for the regulatory things to change, it takes years, if not decades, right? We're just not going to get there. So I think for us to engage earlier with the, you know, with even like the ontology and moving that forward is starting to drive that. I think, you know, one of the things also for the ontology that comes in, so you're looking at electronic health records for people, right? So even hospital, for example, at University of Pennsylvania, they have 7 hospitals, right? And even between the electronic health records have been around for a very long time, but they're not aligned, right? So somebody even mentioned a comment like for a neutrophil, I think there's like 7, or for the white blood cell, there's like 72 terms for it. So how do doctors now agree on that one term, right? So what's the metadata, Stefano, what you brought up? And I think for us, it's definitely important to address. But I think before waiting even for one of those parties and regulators to jump in, we just have to do it together and start moving it forward and engaging, maybe even at a smaller projects or more focused topics. But that's what's going to help us really drive that engagement and expedite that engagement with these technologies, at least from my perspective.
Yeah, thank you, Stefan. I will have time. So one comment from me, if anyone is interested in joining the initiative, please email me for more information. As Shepan mentioned, we're always open to new participants in our group. And so it's great to have people here. I will have time for just one more question. So data quantity, 2 terabytes an hour is quite a lot. How has Recursion and or others managed this limitation? Any learnings to share? So Laura, do you wanna? Yeah.
Yeah, I'm happy to take a crack at that one. So I would say that Recursion, I don't know if it's actually special. I know that a lot of the big pharmaceutical companies and more companies are focused on capturing and collecting data. So I can share openly in this forum that Recursion has more than 10 petabytes of data that we store on a regular basis. So actually, in reality, for us, 2 terabytes an hour is kind of child's play. I can recognize that that might be a challenge for others, but what I would say is that with the kind of continued expansion of cloud providers and the ability to store data at varying tiers of expense. So you can actually take data and put it in what they call like deep cold storage. It's actually incredibly inexpensive. And so, I think that's a huge part of why we talked quite a bit today about engaging with the technology providers, understanding what your cloud solutions are, talking with IT, cybersecurity, et cetera. The reality is collecting that amount of data, if you can route it to the right place, and again, metadata is really important here too, so you can go back and find the data you're looking for. But that if you can route it to the right place, that there are increasingly ways to keep that amount of data available to you in a very relatively inexpensive way. And so that's what I would recommend, is to talk with your IT and others at your companies to see what options are available to you, because it is actually much cheaper than you might expect.
Awesome. Thank you, Laura. I am going to launch real quick a final poll to just ask people to rate the webinar overall. And then I will just remind everyone that this is a webinar series. This is the first of 4, so please stay tuned for the next invitation. We're going to go more into the depth onto practicalities, onto use. We're thinking about having some like Ask the Experts sessions. They should be pretty exciting. If you registered for this webinar, I'll make sure that you get a targeted invitation to register for the next webinars. And hopefully we'll be deciding and releasing those dates soon. So with that, do any of the speakers or Shepan as the lead, do you have any final words for our guests today?
No, I just want to thank all of the speakers participating today and pulling this together, but also the whole team that's part of this initiative, right? Putting the paper together. There's an enormous amount of work that goes behind the scenes. So I just wanted to acknowledge everybody there. And also thank you for folks for attending today. And I hope you and others will join us down the road. Yeah, and I'll pass it on for final comments to others.
Awesome. Well, thank you, everyone. Thank you, attendees. Thank you, speakers. Again, I'll send out the recording in the next couple days, and we look forward to having you on future webinars. Hope you all have a good rest of your week. Okay, bye, everyone.
Cheers, everybody.
Be well.